Next, we're going to look at proteins. Proteins are an organic biological molecule made up of amino acids. So the monomer of proteins are amino acids. All amino acids have a central carbon and then two attached functional groups, one a basic amine group and the other a carboxyl acid group. Proteins are also composed of a combination of 20 different types of amino acids, and they'd be bound together with peptide bonds. If you're talking about a polypeptide, you're probably talking about what essentially is a small protein of 10 to 50 amino acids. We use protein more for macromolecules that contain anywhere from 100 to 10,000 amino acids. Here's what an amino acid looks like. You can see the amine group on the left, the carboxylic acid group on the right, and a central carbon attached to both a hydrogen and an R group. The R group is the variable group, and depending on which amino acid you're talking about, it looks different. Here's a quick schematic that's showing you the diagrams of the 20 amino acids that we find in the human body. Now, some do have to be consumed, so they have to be found in what we eat. They're not made in the human body. And what do proteins do? Well, they have both structural and functional components to the body. They comprise 20 to 30% of cell mass and contain carbon in combination with hydrogen, oxygen, and nitrogen. You may even find sulfur and phosphorus in proteins. They are going to be polymers of amino acids, so they're essentially big molecules made up of repeating units of amino acids held together by peptide bonds, and they actually have 4 structural levels. Some examples of functions for proteins are providing structure to cells, but also acting as enzymes to speed up chemical reactions or allowing for movement like contraction in muscles. To look at the structural levels of proteins, we're going to look at them all together first. The 4 levels are very simply numbered 1, 2, 3, 4, or primary, secondary, tertiary, and quaternary. The primary level of organization is just the linear sequence of amino acids. What order are the amino acids in? The secondary level is when that linear sequence starts to take on shapes, like a helix or a pleated zigzag sheet. That's the secondary structure. Tertiary structure is for some proteins as far as they go, where their secondary structures start to interact and they start to take kind of a globular shape. If you have 2 tertiary structures come together, then you're looking at the quaternary level of organization for a protein. So again, these are just the basic organization levels. It's really important that proteins maintain a certain structure and shape. Structure and shape is important for proteins because they may stop working if that shape changes. So here at the primary level, we're looking at essentially beads on a string, the order of amino acids. At the secondary level, we're looking at shapes of those beads on a string, like a spiral or a zigzag, which are alpha helices or beta pleated sheets. When those zigzags and pleated sheets and helices come together, we're looking at tertiary structure. So that's where we have interaction among those secondary structures, and we start to see more of a globular shape. And some proteins stop here. Some don't make it to this quaternary structure, where you have 2 tertiary polypeptide chains come together to create a functional protein that has a specific shape. So when we're talking about proteins, we're talking about things that are made up of amino acids, have amino acids as their monomer. They're the basic structural and functional material of the cell. They're abundant in cells. And examples include things like enzymes and other important functional proteins like hemoglobin that carries oxygen, or the contractile components of muscles. And their structure and shape is so important that they have 4 levels of structural organization, from primary to quaternary. From where they're just in order to where they take on shapes, where those shapes interact, to where those bigger shapes that interact to form globular molecules interact with each other and aggregate together. So when we're looking at proteins, we're probably looking at either a fibrous or globular protein. Fibrous, those proteins, they tend to be more structural. They're strand-like, they might have tertiary or quaternary structure, their 3-dimensional structure is still important to them, but they have more mechanical and tensile strength in their shape. Keratin, collagen, those are just some examples of fibrous proteins. If you're familiar with proteins, though, you're probably most familiar with globular proteins, like antibodies and hormones and enzymes. Those are going to be functional proteins because they carry out a function like carrying oxygen, protecting us against pathogens or things that cause disease, communication within the body using the blood, or even speeding up chemical reactions. Their shape is even more important to their function than that of fibrous proteins because they're going to have functional areas called active sites where things bind to them in order to make them work or work better. They tend to be more globular and compact. They might have a more spherical shape. A lot of them are water soluble and even very sensitive to environmental changes. So here are basic diagrams showing collagen, a fibrous protein on the left, which provides a lot of structure, and a globular protein, hemoglobin, on the right, which is more functional. Hemoglobin actually carries oxygen and is a 4-subunit quaternary protein. Protein structure is so important that if environmental conditions shift, it may denature a protein, basically make them unfold and lose their functional 3D shape. Fibrous proteins are going to be more stable than the globular proteins, whose active sites can easily become deactivated because that shape change can disrupt those active sites. And environmental changes like increases or decreases in pH or increases and decreases in temperature, especially to extremes, can unwind proteins. Now, in some conditions, this is reversible, but in others, it's irreversible. If you've ever cooked an egg, you've actually denatured proteins. You've changed proteins which had a more liquid state into those that were denatured and took on a solid state. And if you've ever tried, you probably weren't successful in trying to uncook an egg. So sometimes this can be very damaging to the function of proteins when it's irreversible. Take this protein, for example. This one has a very well-defined shape. If you were to unwind it, a lot of its functionality, such as its central active site, would disappear and not allow it to function as it should. One of the most important types of proteins is an enzyme. Another name for an enzyme is a biological catalyst because they can speed up or increase the speed of chemical reactions without getting used up in the process, so they can work over and over again. The way that enzymes speed up chemical reactions is they lower the amount of energy needed to initiate a chemical reaction. They lower something called activation energy. That allows for millions of reactions to happen per minute, and it helps us
An
sustain life.
enzyme is a protein that changes the rate of a chemical reaction without itself being changed into a different molecule in the process. Enzymes promote and regulate virtually all chemical reactions in cells.
As
An
you've seen in the
enzyme is a
video,
protein
there's
that
actually
changes the
multiple
rate of a
steps
chemical
involved
reaction
in enzyme
without
action,
itself being
such as
changed
a
into a different
substrate
molecule in the
binding to an
process.
active site, forming a
Enzymes promote
substrate
and
enzyme
regulate
complex,
virtually all
and then
chemical
rearranging
reactions
the
in cells.
reactants to produce a product. But the enzyme
An
is not going to
enzyme
be
is a
used up
protein that
in this
changes the rate
reaction.
of a chemical reaction
Examples of enzymes
without itself
include
being changed
names that
into a
end
different molecule
oftentimes
in the
in ACE,
process.
like hydrolases,
Enzymes promote
oxidase,
and
lipase,
regulate virtually
catalase.
all chemical
Enzymes are
reactions
very
in the
specific and
cells.
act only on specific substances called that
An
substrate.
enzyme is a protein
And so
that
we're going
changes
to
the
see
rate of
more
a chemical
examples of
reaction
how biological
without
catalysts
itself
or
being changed
enzymes
into a different
lower
molecule in the
activation
process.
energy. So here we have two charts, one on the left and one on the right. The one on the left is showing how reactants get turned into products without an enzyme present. You can see the hill that the reactants have to overcome, the amount of energy required to produce the product. It's a lot more without an enzyme. With an enzyme, the amount of energy or activation energy required has lessened because enzymes lower the activation energy by bringing reactants together to more likely increase interaction and produce a product. So enzymes are biological catalysts because they catalyze reactions by speeding them up. They speed them up by decreasing activation energy. So here's an enzyme in action. We have the enzyme, we have its active site where something can bind, it has a particular shape. We have substrates that are specific to an enzyme and bind to the active site. And here, these substrates are two, and they're going to come together and be held close together to form a bond once we have the enzyme substrate complex. So what's released as the product is going to be a more complex molecule than what we began with. In this example, we're actually making a peptide bond because we're combining two amino acids. So we're building a really small polypeptide. And last but not least, we're going to look at nucleic acids. Nucleic acids are in all the food we eat. They contain carbon in combination with hydrogen, but also oxygen, nitrogen, and phosphorus. The monomer for nucleic acids is nucleotides. And nucleotides themselves are composed of three things: nitrogen, a pentose sugar, or a 5-carbon sugar, and a phosphate group. And repeating units of these nucleotides can build the two major classes of nucleic acids, which include DNA, deoxyribonucleic acid, and RNA, known as ribonucleic acid. When we're looking at nucleic acids, we're looking at nucleotides. And in DNA, they can have 5 different nitrogen bases. They can be made up of adenine, guanine, cytosine, thymine, or if we're looking at RNA, uracil. Uracil is only found in RNA, whereas the others, guanine, cytosine, thymine, and adenine, are found in DNA. When you're talking about RNA, use sub uracil for thymine. DNA is a double helix. It has the sugar and phosphates of its nucleotides on the outside of the molecule, with the bases in the middle. It's able to replicate itself before a cell divides and ensures genetic continuity among parent cells to offspring. DNA is also a genetic blueprint, and it provides information for how to synthesize all the proteins of an organism. It contains within it deoxyribose sugar, and that is different from the sugar we find in RNA bases. For DNA, we're looking at, again, a double helix with the bases adenine, thymine, guanine, and cytosine. And the way the two strands pair together is by their bases. And there's complementary base pairing rules, where A always pairs with T, or adenine always pairs with thymine, and guanine always pairs with cytosine, or G always pairs with C. Ways to remember that is the apple fell off the tree, and the car goes in the garage. There's other phrases to remember, but those are the ones I use. Here's the basic structure of DNA. You can see at the top 2 different nucleotides, an adenine nucleotide on the left, a thymine nucleotide on the right. Both nucleotides are made up of a nitrogen-containing base, a sugar, since it's DNA, it's deoxyribose, and a phosphate group. The sugar and phosphate groups make up the backbone of DNA, and the bases, which are shown in a colorful array down at the bottom image, make up the middle, or kind of like the rungs on a ladder. What holds the bases together are multiple hydrogen bonds. Whereas RNA is single-stranded, DNA is double-stranded. RNA is a single-stranded molecule that's found in both the nucleus and in the cytoplasm of a cell. DNA is only found in the nucleus of a cell. So we need RNA as a go-between molecule that can take the genetic instructions from DNA to other areas of the cell. RNA contains ribose sugar, not deoxyribose like DNA. Instead of using thymine, it has uracil as a base. And there's actually 3 varieties of RNA. Messenger RNA, which is the real go-between molecule between the nucleus and the cytoplasm. Transfer RNA, that deals with moving amino acids around to build proteins. And ribosomal RNA, which makes up a ribosome, which is a protein-making machine in a cell. The nucleic acids in RNA, again, just to make sure you have them separated in your mind from DNA, is guanine, cytosine, uracil, and adenine. Almost the same as DNA, except RNA has uracil instead of thymine.
DNA and RNA are nucleic acids, polymers made of subunits called nucleotides. One difference between DNA and RNA is the type of sugar their nucleotides contain. DNA contains the sugar deoxyribose, while RNA contains the sugar ribose. Ribose has one more oxygen atom than deoxyribose. DNA and RNA are each composed of 4 different nucleotides, which differ in their nitrogenous bases. 3 of the 4 bases are the same in DNA and RNA: adenine, guanine, and cytosine. The 4th base in DNA is thymine. In RNA, it is uracil. The nitrogenous bases guanine and adenine each have 2 linked rings of atoms. They are called purines. Cytosine, thymine, and uracil each have a single ring, and these 3 bases are called pyrimidines. For convenience, the carbon atoms in a nucleotide sugars are numbered, beginning with the carbon atom bonded to the nitrogenous base, moving around the ring, and up to the carbon that is bonded to the phosphate group. The 1' carbon is bonded to the nitrogenous base, the 3' carbon to the next nucleotide, and the 5' carbon to the phosphate group. DNA and RNA are polynucleotides, long chains of nucleotides. Polynucleotides are always assembled in the 5' to 3' direction. A covalent bond forms between the carbon at the 3' position of a nucleotide and the phosphate group at the 5' position of the next nucleotide.
As you can see in that video, there's a lot of information to know about DNA and RNA, and we'll talk more about that when we get into a chapter that deals with cells and cell machinery. But to finish up talking about chemistry and biological molecules, we want to look at adenosine triphosphate. Adenosine triphosphate can be abbreviated as ATP, and it's a source of immediately usable energy for the cell. It's essentially like energy currency for the cell. What ATP looks like is 3 phosphates attached to essentially the rest of a nucleotide. So you would have an adenine nitrogen containing base and a sugar. ATP has ribose, adenine, and 3 phosphate groups. And it's those last 2 phosphate groups that have high energy phosphate bonds that can be broken to release energy, allowing cells to utilize that bond breaking energy. Here we can see that you can both break down and build ATP. To break down ATP, you add water in a hydrolysis reaction to break it down into adenosine diphosphate, or ADP, and inorganic phosphate, and that releases energy. You would need energy to combine ADP and inorganic phosphate together to build ATP again, and you would utilize a dehydration synthesis reaction or removal of water to make that bond. So like many reactions, building and breaking down ATP is a reversible reaction. But when ATP is made, even though it's energy expensive, it provides so much more to cells, like the ability to carry out transport work, mechanical work, and chemical work. The ability, for example, to transport chemicals across cell membranes, to contract different types of muscle cells, and in order to phosphorylate reactions that allow for energy absorbing chemical reactions to take place, such as metabolism in the human body. So keep these things in mind as you further your study of chemistry and you look at biological molecules more, you formulate questions, and you prepare to go on to a chapter all about cells and eventually learn about organ systems.